Hi, I'm Kevin Esoh, and I'm pleased to share findings from our review on the evolutionary history of sickle cell mutation. The sickle cell mutation is a single nucleotide change in the beta globin gene, which results in hemoglobin structural change. When inherited from both parents, it causes sickle cell anemia, which, if not diagnosed early in life and treated, can claim the lives of up to 90% of all affected babies. Ideally, such a mutation would be strongly negatively selected and purged from populations. Why then has a sickle cell mutation persisted? The first comprehensive explanation to this puzzle was introduced in 1949 by Harding, who estimated that the beta thalassemia trait conferred a gain in fitness against severe malaria in the Mediterranean region, where both conditions were prevalent. Harding's malaria hypothesis was confirmed by Anthony Allison in 1954, when he observed in Africa that the sickle cell trait conferred protection against severe Plasmodium falciparum malaria. Based on this wide distribution and specific haplotypes on its genetic background, the origin of the mutation was greatly contested. It was not until 2018 when doctors Daniel Schreiner and Charles Rotimi confirmed the single African origin of the mutation. However, the model for their age estimate assumed that the viability of homozygous individuals was 0 instead of 20%. In 2019, researchers from France used accurate viability estimates and proposed that the mutation arose about 22,000 years ago. However, their model underestimated the equilibrium frequency of the mutation. Therefore, more research is needed to determine the true age of the mutation. Interestingly, both groups agree that the mutation arose in the genetic background of agriculturalist populations. Based on haplotype analysis, Schreiner and Rotimi suggested that the mutation arose somewhere along the border between Cameroon and Nigeria. Indeed, data on the origin of Plasmodium falciparum suggests that the mutation arose somewhere in Cameroon. What's the implication of studying this evolutionary history? Theoretically, in an evolutionary neutral environment, no affected baby should survive sickle cell anemia. However, in reality, in malarial environments, up to 20% of affected babies can survive to adulthood undiagnosed and without treatment. This long survival of treatment-naive sickle cell anemia patients has been linked to biological factors, some of which are highly heritable, such as fetal hemoglobin level. The genetic factors that influence these traits may be ideal targets for therapeutic approaches. This is an area of particular interest and therefore the focus of research by the Sickle in Africa Consortium. Also, the interaction of the sickle cell mutation and other gene variants under adaptive selection in sub-Saharan Africa can either increase or decrease sickle cell disease severity. Therefore, the management of sickle cell disease must take into account these comorbidities. Thank you.